Today we are going to be looking at Chapter 4. This chapter is looking at how life is cellular. So we are going to focus on cells, specifically their structure and function. In your book, they tell a story about how some scientists are trying to build artificial cells from scratch using the knowledge that we have today based on research that had been done in the past. And I'll point this out when we get to the cell theory. If we go back in Chapter 1, we looked at something called the biological hierarchy. And this is showing the different levels that we can study within biology. We spent some time looking at atoms and molecules in the previous chapter. These atoms come together, they form bonds to create molecules. Molecules include water, carbohydrates, proteins, lipids, and nucleic acids. Those biological molecules, along with water, will come together to create cells. So this is our next step up within this hierarchy that we are going to look at. Cells are defined as the smallest and most basic unit of life. In addition to being the most basic unit of life, these cells are microscopic. So we have to use microscopes in order to view them. And we have many different kinds of microscopes that can be used to study cells. And these microscopes are very powerful. So with these microscopes, we can study the cells and we can see that they are self-contained units that are enclosed by a protective membrane. And here we have some examples of different types of cells. Up in the top left-hand corner, this is a bacteria cell called Salmonella. In the top right-hand corner, we have a plant cell over here, and you can see that there are many different colored structures within the cell. The bottom left picture shows a yeast cell, so it has a lot of structures in it as well. And the fourth picture in the bottom right, these are human nerve cells. And this is to demonstrate that cells can be specialized, they can have special structure, which will lead to a specialize in function. All of these cells that we're going to be looking at, they fall into a theory called the cell theory. This cell theory consists of two main parts. The first part states that every living organism is composed of one or more cells. This means that we can have organisms that are unicellular or single-celled, such as bacteria. We can also have organisms that are multicellular, or they have multiple cells in them, such as you. So you have approximately 10 trillion cells in your body. And some people even estimate that it is 100 trillion cells in our body. The second part of the cell theory states that all cells living today came from pre-existing cells. And this happens through cell division. We'll look at the two types of cell division in a later chapter, but they are mitosis and meiosis. The second part of the cell theory, this is the part that scientists are looking at, and scientists are trying to make cells that are artificial that did not come from pre-existing cells. And they've had a little bit of luck, but not quite enough, as you can read in your textbook. There are two different types of cells that we will be looking at, and in both of these types of cells, they will contain DNA. And DNA, we talked about this a little bit in the previous chapter. DNA is deoxyribonucleic acid. The functions of DNA include that it acts as a set of instructions for the cells. Those sets of instructions encoded for in the DNA is also passed down from parent to offspring. So DNA is very important for giving you all of your traits and characteristics. It's also very important because it passes down to offspring. And it does that in order to sustain life from one generation to the next. And as we learned, DNA is made up of building blocks called nucleotides. And those nucleotides will bond together and form a structure called a double helix. So here we have a picture of DNA on the right-hand side of the slide. And we have two strands of DNA in the blue, and their nitrogen bases will face in towards each other. As those nitrogen bases attach to each other using hydrogen bonds, they will form a twisting helical structure. All of the DNA inside of a cell is referred to as the genome. So we, for example, have sequenced the human genome. So this will include the cell's complete set of genetic information. If we zoom in on the genome, so here we have our genome here in the cell. If we zoom in on that DNA, it's organized in chromosomes. Those chromosomes, if we extend them out to the DNA molecule, within those DNA molecules, there are units referred to as genes. So a gene is a segment of DNA that codes for a distinct genetic characteristic. Specifically, these segments of DNA, these genes, will code for specific proteins. So that's really important. So here we have two of our biological molecules that we have looked at. We have the DNA, we also have proteins. And DNA, they encode for those instructions to create proteins. And remember, proteins have very diverse functions. So some of these proteins will be used for transport, some proteins will be used for movement, some proteins will be enzymes, etc. Another big characteristic besides DNA that cells will share is that all cells have a plasma membrane. And a plasma membrane, this is a barrier. It's made up of phospholipids. So here we have one of our lipids that we looked at in the previous chapter. And these lipids, they're going to separate the cell from its external environment. So you can think of this as the skin of the cell. So looking at these phospholipids have two parts. They have a hydrophilic head. This means that that circle head region is water loving. Attached to that, we have two hydrophobic fatty acid tails. And these are water fearing. So if you take a bunch of phospholipids and throw them into water, they will orient themselves into a double layer, or it's called a bilayer. So we'll have phosphate heads facing out on either end of the membrane layer, and those fatty acid tails will face in because they're hydrophobic. They're trying to avoid being by water. This plasma membrane, besides defining the cell, another big thing is that it is selectively permeable. So this means that some substances can pass through the membrane very easily, whereas other substances do not pass through. And being able to pass substances through the membrane is very important because cells can bring in good things like nutrients or water. And cells are also able to get rid of bad things like waste products or toxins. So in order to take advantage of the plasma membrane being selectively permeable, there are different types of transport to move these substances across that membrane. The first type of transport is called active transport, and this is movement of a substance across a membrane. And I'm going to cross this out because this is wrong. So this is when we're going to move substances across a membrane from low concentration, so where that substance is less common, to high concentration, where the substance is more common. So in order to do this, this requires a transport protein that will be embedded into the plasma membrane. and that is this orange structure here. In addition, we also require energy in the form of ATP. And this process is not spontaneous. It requires energy, it requires a protein transport molecule, because when we move molecules from low to high concentration, this is going against what the universe naturally wants to do. Usually substances will spread out or diffuse. They'll be going from high concentration to low concentration, or they'll try to get to equilibrium. Active transport, you can think of it as we are trying to create order, or we're trying to pick up those molecules and place them in a spot. See, another way to think about this is naturally in your bedroom, things will go towards disorder. Things will get messy. So what you have to do in order to make your bedroom clean or to reorder things, you have to put in energy, you have to put in work. So that's what active transport is doing. It's trying to organize things, put things closer together into a high concentration. The other type of transport is passive transport. And we're going to look at a couple of different types of passive transport. So passive transport is when substances move across a membrane without any additional energy. So this is when molecules will go through diffusion. And diffusion, this is a movement of substances from an area of high concentration of that substance to an area of lower concentration of that substance. And this is how the universe works. Molecules tend to spread out, or they tend to diffuse. So because this is how the universe works, we don't have to invest any energy in order for this to occur. Or another term is spontaneous. So this is how molecules will spontaneously move. They'll try to spread out and get to equilibrium. So here we have an example. If you have a glass of water and you place a food coloring drop in there, that food coloring over time will spread out until it's at equilibrium, or it's evenly spread out in that glass of water. A type of diffusion that occurs across a plasma membrane is simple diffusion. So this is when we have direct diffusion through a membrane, so no energy is required, it is spontaneous, and there's no transport proteins that are required. An example of this is shown in the image here with water. Water molecules are fairly small. They have 3 atoms to it because they're H2O. They have 2 hydrogens, 1 oxygen, so 3 total atoms in that molecule. Because water is so small, it can pass through that plasma membrane, or it can use simple diffusion to go from high to low concentration. Other molecules are larger, or they're ions, so things like sugar or potassium ions, they cannot use simple diffusion to go from high to low concentration. Instead, they use facilitated diffusion. Facilitated diffusion, this is diffusion through a membrane that requires a transport protein. But again, because it is diffusion, there's no energy required, it's spontaneous, it just has a helper to help that molecule move across from high to low. And there's a couple of examples of facilitated diffusion. So potassium ions, there are sugars that are larger molecules. Here we have calcium, which is also an ion. And it's still going from high concentration to low concentration. A special type of simple diffusion is osmosis. And osmosis is when we have water molecules moving from areas of high concentration to areas of low concentration. And this osmosis, this is going to set up different environments for our cells. And those different environments are referred to as tonicity, and there's 3 different types of environments. We'll start with the middle one here, which is called an isotonic solution. An isotonic solution is when the amount of water outside the cell is the same as the amount of water inside the cell. So this means water is still moving in and out of the cell, but at the same rate. Iso means equal, so water is equal inside and outside of the cell. In a hypertonic solution, over on the left-hand side, a hypertonic solution means that there is less water outside of the cell. There's more water inside of the cell. So we have a high concentration inside the cell. That means water will flow out. Water is going to go from high concentration in the cell to low concentration out of the cell. In a hypertonic solution, when water flows out of the cells will shrivel. And we see that in our animal cell at the top and our plant cell at the bottom. They're both shriveled in this hypertonic solution. In a hypotonic solution, it's the opposite. This means there is high concentration of water outside the cell. There's low concentration of water inside of the cell. Water is going to go from high to low, so it's going to flow into the cells. In animal cells, if too much water flows into the cell will burst open and die. In plant cells, plants really like this type of environment. Water will flow into the plant cell, but plant cells have a cell wall around them that prevents too much water from flowing in. So plant cells will fill up with water, they will be very turgid or rigid, and this will allow plants to stand up against gravity. If you look at the animal cells, they like isotonic solutions. So animal cells do not want to shrivel up. They also do not want to fill up and burst with water. So different types of solutions that we place cells into can influence what our cells look like or the structure of them and also how they function. Another type of transport that can occur for cells happens when cells create vesicles. So vesicles are when a cell can form a secretion, or formed when sections of a plasma membrane, they will bulge inward or outward to form spheres. If these spheres contain something that the cell wants to get rid of, those vesicles can travel to the plasma membrane of the cell and do exocytosis. So this is when we're going to export a substance out of that cell. Other times cells will want to bring substances into the cell, and this is referred to as endocytosis. And there's two types. There's nonspecific endocytosis, when all the material in the area is surrounded, enclosed, and imported. And this can happen when the cell does phagocytosis or panocytosis. So when the cell brings in food, like bacteria cells, or when a cell brings in a liquid with solutes dissolved into it. Another type of endocytosis is specific endocytosis. This is when one specific molecule is surrounded, enclosed, and imported. An example of this is receptor-mediated endocytosis. So on the outside of the plasma membrane, the cell will have receptors that will grab that specific target molecule. Once that target molecule is attached to the receptors, that plasma membrane will fold in and create a vesicle, and it will bring that molecule into the cell. So there are two types of cells. There are prokaryotic cells and eukaryotic cells. Both of these types of cells have DNA, or they have that genetic material. In addition, they have a plasma membrane. So we're just looked at similarities between those two types of cells. Besides having similarities, the two types of cells also have major differences. So I'm going to write those over here on the slide. So prokaryotic cells, this one up here on the top. Prokaryotic cells are small. They are also fairly simple. They are defined as lacking a structure called a nucleus. In addition, prokaryotic cells are found in 2 out of our 3 domains. They are found in the bacteria and the archaea. So those are a few characteristics that prokaryotic cells have. And now we can compare those characteristics to our other type of cell, which is the eukaryotic cell. Eukaryotic cells are larger and more complex. They have a structure called a nucleus, and this is where that DNA is found. And eukaryotic cells are found in our third domain of life, the eukarya domain. So we can see that these two types of cells, they do have a few differences. We are going to really focus on the eukaryotic cell because it is more complex. We're going to be looking at the different parts within that eukaryotic cell and their functions. Prokaryotic cells are simpler. They really don't have a lot of structures inside of them. They will have DNA in a nucleoid region. They have a plasma membrane. They may have a cell wall outside of the plasma membrane. They have ribosomes that will create proteins. So they're very simple structures. And we'll see some of those structures when we get through the eukaryotic. So we're going to start off with our eukaryotic cell. They're larger, more complex, and they have a nucleus. So that nucleus, that is the control center of the cell. And it contains structures called chromosomes that are made up of proteins and DNA. That DNA is that genetic material. The proteins that are associated with it, they just help to organize that DNA into these chromosomes. The nucleus has a nuclear envelope. So this is what makes it a separate structure, is that it has a double lipid bilayer membrane that forms the boundary of the nucleus. It's very similar to the plasma membrane, so plasma membrane defines the cell. The nuclear envelope defines the nucleus. That nuclear envelope does have pores in it, and these are channels that allow products to go in and out of the nucleus. So just like our plasma membrane, the nuclear envelope is selectively permeable. Things can go in and out of the nucleus depending on what the cell needs to do. The next structure we're going to look at is the endoplasmic reticulum, or ER for short. This is also a lipid bilayer membrane network. It's continuous with the nuclear envelope, so they are attached to each other, but they have different functions. There are 2 types of ER. There is smooth ER. This manufactures lipids and hormones. It can also break down any toxic materials, such as alcohol. The second type of ER is rough ER. The rough ER is covered with ribosomes that make proteins for export from the cell. So in short, rough ER makes proteins, smooth ER makes lipids. So again, we see our different biological molecules showing up within our cells. Our next structure is the Golgi apparatus. This is a packaging center that directs those proteins and lipid products that are created in the ER to other membranes. You can think of the Golgi apparatus as a post office. It's going to package, it's going to tag and ship out those products that were created in the ER. Some of the vesicles coming out of the Golgi apparatus are lysosomes. So these are a lipid bilayer membrane enclosed sac that breaks down biomolecules such as proteins into simpler compounds that can be recycled and reused. You can think of the lysosome as the recycling center for the cell. So lysosomes are going to break down anything the cell no longer needs back into its building blocks that can be used to create other molecules. Another structure are the mitochondria. Mitochondria are found in both animal and plant cells. And these structures, they use chemical reactions to transform energy of food molecules into ATP using the process called cellular respiration. And we will come back to how this process actually works in the next chapter. In addition, cells will have a cytoskeleton. This is a network of proteins. They're in cylinders or filaments that provide structure to the cell. So there's a couple of different functions of the cytoskeleton. One is to form the framework of the cell. Another is to organize the interior of the cell. So it's going to organize all those different organelles, the Golgi apparatus, the mitochondria, etc. The cytoskeleton is going to support the intercellular movement of those organelles. And cytoskeleton in some organisms enables the whole cell to move, such as in amoebas. The cytoskeleton also gives shape to cells that do not have cell walls outside of their plasma membrane. There are two other structures that we're going to look at within our eukaryotic cells, and these two structures are found in plant cells, but not in animal cells. The first structure is called a vacuole, and this performs similar functions to those of the lysosomes. So they help to recycle nutrients, and it also helps to store water for physical support of the cell. And this goes back to that osmosis that I mentioned earlier in the chapter. So plants really like to be in those hypotonic environments where water flows into the plant cells. And that water that is flowing in is going to be stored in the vacuole. Some vacuoles will stockpile noxious compounds that will be released when a herbivore eats a plant. So they also perform storage functions. In addition to the vacuole, plant cells also contain chloroplast. And chloroplast, this is an organelle that will capture energy from the sun and use it to manufacture sugar via the process called photosynthesis. And we will take a look at photosynthesis in chapter 5 along with cellular respiration. So these two types of cells are prokaryotic and eukaryotic cells. They have similarities to them. They both contain genetic material in the form of DNA, and they both contain that plasma membrane that defines the cell. But then we also looked at some differences. The prokaryotic cells are simple and smaller. They do not have a nucleus. They're found in the domain bacteria and archaea. The eukaryotic cells are bigger, more complex. They do have a nucleus along with other organelles, and they are found in that third domain, the eukarya domain.